So the first ever collective behavior meeting that I think ever happened, that I was aware of, was here at IPAM a very long time ago. So it's great to be back all these years later. And I want to talk about cognitive collectives. And so what are my interests? Well, I'm really interested in computation. And so what goes on inside the brain of individuals and how the invisible network of communication between individuals give rise to higher forms of collective intelligence. And we've already seen these beautiful movies before. This is a sort of really fascinating example of this type of process. So I want to talk about the connection between individual and collective decision-making, and more broadly about spatiotemporal computation in natural evolved systems. So let's start off with the earlier models that we've heard quite a bit about earlier today, which speculated that individuals are interacting locally with neighbors, such as to align with nearby individuals. And if we scale that up using computer simulations, we're able to show patterns that look reminiscent of what we see in nature. And around that time, that was kind of all we had. We didn't have good experimental data for these systems. We didn't have AI-based tracking tools that we have today. And so in this particular model, which is now known as the Couzin model, we have the zone of repulsion here, a zone of orientation, and a tendency to be attracted to neighbors. And as you saw, it looks as if it's quite convincing in terms of explaining group-level properties. Another model, which has preceded this, which is highly influential, is called the Vicsek model. Again, those of you who've been here all day today have heard a lot about this model too. And one of the beautiful things about this model is it just assumes that individuals align their directions of travel with near neighbors, and this could lead to large-scale macroscopic properties. So Tamas kindly shared me the video of his very first animation of this model. And this is what really captivated people's attention, because again, it's producing patterns and properties that look like the types of groups we see in nature. Now, if you change the density within the Vicsek model, so this is just a different rendering, you can see it's largely disorganized, but as we increase density further and further still, these little noisy agents are then sort of averaging the information with those around them, and we get what's called the density-dependent transition from disorder to order. So if I just show this again from the beginning, you can see that at this low density, individuals are pointing in all sorts of different directions. As density increases, you get this spontaneous kinetic phase transition into an ordered state. And it looks like flocks and schools. It looks like the types of groups we see in nature. And this just quantifies, as you increase the density here, you undergo this kinetic phase transition from disorder to order. Order here is the alignment of the system in a global sense. Now, back in the early 2000s, we were thinking, well, this is really fascinating. Let's go to nature and look at animals like locusts, which are a global plague that I'll come back to at the end of the talk, and ask whether the Vicsek model explains reality. And so this paper was published in 2006, and it's been very highly cited because it drew that connection between these different scales of organization. And so what we did in the lab was we set up this annulus arena where locusts would march around the arena. They're not the smartest animals out there. They think they're in a never-ending desert environment. And I developed some of the first software to track the individuals so we can get quantitative data for long periods of time, because the locusts march for about 8 hours a day. And so if we think about this again, the Vicsek model, if we put that into an annulus arena, we can get this type of collective properties that we see in our experiments. And to calculate the alignment, the order, of course, because we're in a rotational system here, we can't just add those vectors together, because then it would be actually appear to be disaligned. But what we can do is we can calculate the alignment with respect to the center. So in this case, they're mostly marching counterclockwise, so that would be a negative value, maybe around negative 0.8. If they were marching clockwise, that would be a positive value. Oops, am I going the right way? Sorry. And so then if we look at the 8 hours of our experimental day, and we just look at the alignment, as I said, calculated either negative or positive, you see it looks kind of dense, it looks kind of disordered for these low densities. And as we increase density here, you can see the locusts were marching counterclockwise in this particular example, and then they show intermittency, they switch to clockwise, and so on. And at high enough densities in this particular experiment, for 8 hours they marched clockwise. And so we thought that this was reminiscent of what we saw within the theory, the Vicsek model, whereas at low densities you have this gaseous-like phase, and then you get this kinetic phase transition from disorder to order as a function of density. And perhaps also compelling, or seemed compelling at the time, is that if you look at the total time spent in the ordered phase, so in that paper we said anything above 0.3 was considered ordered, you can see again, as you increase the density of the locusts, you have this nonlinear increase in the order of time spent ordered within the system. And this is the theoretical predictions, and these are the experimental data. So again, it all seems self-consistent and seems to support the Vicsek model as being a foundational model for collective behavior. And as I mentioned before, we calculated the alignment. But recently we've realized that there's a slight problem with that, in that the alignment itself, the calculation, can be dependent on the number of individuals that you actually utilize in the calculation. So to make it a fair process, what we can do now is we can calculate just pairwise alignment between individuals. We can randomly take 2 individuals from this large density and just calculate what's their alignment with respect to each other. And the way in the original paper it was done was you had to have a minimum of 2 individuals to be moving to consider them to be aligned. If they were not moving, if no individuals were moving, then the system was considered to be disordered. Now, if we calculate the pairwise alignment, so this is now new data in a paper that's been provisionally accepted but is not published, so it's under embargo. If you actually calculate that pairwise alignment as a function of the number of locusts within the arena, from 120 being the most to 5 being the least, you can see clearly that there doesn't seem to be any evidence of a phase transition from disorder to order. In fact, at lowest densities, if anything, they're slightly more ordered. And this is now just comparing a random 2 individuals taken from here versus here. And another thing to know about locusts is they actually spend around 50% of the time, 52% exactly, but 50% of the time inactive. So if we now think about, well, how on earth can we reconcile that previous figure that showed no density dependence with relation to order, and this figure here where there looks as if there's a dependence? Well, it relates to what I just described to you, that locusts, if this is the order of an individual locust in the population, they spend most of the time ordered with others, but some of the time disordered, around 20% or less disordered. And here, if you look at the probability of the order for any individuals within the population, you can clearly see that it looks remarkably similar. And as I said, the locusts are spending around 50% of the time not active, so they would be considered disordered, and only a very small amount of time when they're active are they disordered. Most of the time, they are fully ordered. So the problem with this analysis is we had no null model. If you imagine you've only got 5 individuals there, and there's roughly a 50% chance of them not moving, sometimes if you flip that coin, you're going to get only 1 head, right? So only 1 locust is moving. That would be considered disordered. No locust moving, considered disordered. Then if you did have 2 flips, there's a small probability that each is disordered with the other, but it's not very high. But if you get to 120 locusts, the probability of having no individuals active is infinitesimal. So if you just do that simple binomial model of what you would expect this to look like with no phase transition, no density dependence whatsoever, you get something like this. So it really was an illusion. There is no density dependence in those data. And so this argument, this key prediction, is that as the density increases, there's a rapid transition from disordered to a highly aligned collective motion, is not supported by the data. So these are the same data that we published there. And so really, the Vicsek model, to my knowledge, this is the only paper that provided empirical support for that model. And now we know it doesn't provide support for that model. And now we can look at data from the field, for example, And you look as if there's maybe some sort of density-dependent transition, but again, there's no null model. What does the null model look like? Because the sample size is different at low densities and at high densities. And again, the null model looks very similar. So, in all honesty, we were so enamored by the Vicsek model and so drawn in by statistical mechanics that we kind of saw perhaps what we wanted to see in those data. And I'll come back later on as to how and why we've completely changed our perspective, because we actually tried to replicate these experiments in virtual reality and couldn't do so. So I'll come back to that. I'll come back to the locusts at the end. Okay, so I'm not trying to be critical of the Vicsek model. It's a beautiful model. It's a really important model. But to my knowledge, it's not supported by any data, at least for animals. Okay, so now let me be critical of my own model. So it's not just the Vicsek model, it's the whole class of models that I have a problem with. And so in this model, there's a key component here, this zone of orientation. And that's what we also saw within the Vicsek model, this tendency to explicitly align with your neighbors. And we saw today in many of the talks, I'm really, I feel privileged that people are still mentioning this model. But let's think about what the evidence is for this. Well, we've known since 2011, where we really looked for alignment among individuals, that there is no evidence of alignment. A fish has the same turning force to an individual that it's moving this direction, and many other analyses that we've conducted, and not just us, that we published back-to-back with a group in Australia at the time, Teddy is now at Cambridge, where they also found no evidence of alignment. In fact, I'm not aware of a single paper where anyone has shown convincing evidence of explicit alignment within animal groups. So if the Vicsek model isn't explained by any biological system, and the Couzin model, this basic premise of alignment has never been found, we're on kind of shaky foundations here for the field of collective behavior. So what I want to argue today is that we've really been thinking about it all wrong. And we actually can get inspiration from going into the brain and trying to understand how do animals navigate through space? And what is known about the neuroscience here? Because we took these models because we were inspired by chemistry and statistical mechanics, but it's completely divorced from cognition. So let's think about what's going on in the brain. And before I go further, I want to thank the people I worked with on this. So Vivek was a PhD student with me and did the simulations and the fly experiments. He's starting a professorship at University of Washington next year. Liang Li is a group leader. He did the virtual reality experiments with zebrafish. Dan Gorbanos was a black hole physicist who joined the lab and did all the analytic theory. Bianca is an undergraduate who did the Locust experiments. And it's a long-term collaboration with Nia, who should be here, but unfortunately, he cocked up his visa application, and Mate Naj, and we've seen several examples of Mate's influence with correlation analysis earlier today. And the really exciting thing is in the last few years, in the case of the zebrafish, literally within 2 years, in the case of flies in the last sort of 5 to 10 years, it has been discovered that this has evolved completely independently, but it's been discovered that they each share what's called a ring attractor of neurons within the brain that they use to coordinate spatial navigation. And so there's a huge amount of interest in this. And we too have these ring attractors in our brain. We know which direction our head is pointing because the brain has to come to a consensus to decide that. So we thought, well, instead of starting with these models the way we just guessed what the interaction rules might be, and we saw that they produced patterns that looked like reality, why don't we start from neurobiological principles regarding the navigational circuits that animals actually use when they move through space? And this is literally how the structure looks in the fly brain. Isn't it amazing that there's this sort of donut structure in the brain? And what's been shown is that the bump of neural activity, this is the colors here just show the different neural groups, but there's a bump of neural activity on this torus that dictates the vector, so the direction and the strength of preference with respect to where the animal wants to go next. I think this is absolutely phenomenal. And as I said, just been discovered also in the vertebrate brain in the zebrafish. So I'm going to talk about individual decision-making for most of the rest of the talk, and then come back to how this tells us something new about collective decision-making. But for those of you interested in collective intelligences, everything is collective, because now we're thinking about the neurons as a collective dictating what the individual does. And this is a basic question. All animals at some point in their lives have to make decisions on the move. Whether you're a locust, whether you're a primate. And so we asked, is there a common decision-making algorithm? And in fact, when we did this work, when we published this, they hadn't even discovered the ring attractor in the vertebrate yet. But you'll see from the data why we had strong confidence that it must be there. And so the theoretical model is so simple. We just assume that there's local excitation, a common motif in neuroscience, and long-range inhibition within the brain. You can also have global inhibition. And we're not the first people to think in this way. This neural ring attractor network that was worked on extensively in the mid-1980s by Chaim Sompolinsky in Jerusalem. But what we've done is a new formulation and also considered as an embodied process, which I'll explain in a moment. So here's an image from experimental data, not ours, from Vivek Jarayaman at Janelia, showing this bump of activity which explicitly codes a vectorial preference within the brain. So we know this is how the animal does it. You can use optogenetics to stimulate another bump, and you can turn the animal in doing so. Okay, so let's take this general framework and consider it in a computational model. And the important thing that we add is that people before have considered decision-making in a very static, non-dynamical sense. Here, what we're considering is as the animal moves through space, that is going to change the geometric representation of the two options in this example, which is going to change the neural consensus dynamic, which is going to change the bump, which is going to influence where the animal moves. As it moves, that changes the geometry slightly, which changes the consensus dynamic, and so on, in an embodied process. And what the model predicts is that the brain exhibits a dynamical bifurcation related to that geometric property. So the system doesn't need to be tuned in any way. The geometry is an inevitability in terms of moving through space. And so here I'll show you the neural activity. And this is just one decision that I'm repeating again and again. You can see the brain chooses the average between the options, and then it reaches this bifurcation point where it undergoes a phase transition and chooses one or other option. In this case, it's always choosing the right, but it's 50-50 if there are equal options. Now, what happens after the animal has reached the target? I'll come back to this later. The animals will tend to oscillate between the options. Right? You wouldn't get that in typical models. But what happens if you put a fly between two options? Well, it will just oscillate back and forth. Right? It allows the brain to break symmetry. You don't want to be stuck in the middle. So this is called Buridan's paradigm. If Buridan's ass is both hungry and thirsty, and there's water to one side and hay to the other, the worst thing for the brain is to get stuck in indecision. So the dynamics of the model allow you to break indecision. We can also take the model to the mean field limit and actually analyze the nature of these bifurcations. And so this is the neural noise, so it's equivalent to temperature in a statistical mechanical system. And this is the egocentric angle between the options that induces this phase transition in the brain. And we can prove that once the animal's brain hits this solid line here, it has to transition and bifurcate into the decision-making state. This is the tricritical point above which there's a second-order phase transition, below which is the first order. And what you're seeing here in this gray region is a hysteresis regime. And actually, the neural noise we estimate from experiments is about 0.25, so they're actually in this region here. What this means is once they hit this dotted line, probabilistically, the brain will transition to a decision. And it can transition anywhere here, but if it doesn't transition by this point here, it has to transition. And because it's hysteresis, once the animal makes a decision, it's very unlikely to flip back to indecision. And so we predict the animal doesn't actually slow down, but the animal will move through a critical point that's defined by the time-varying geometry. So the smoothly varying parameter here for the phase transition is geometric, is the angle. And then it moves towards the target. So why do you think this is important? Well, we think it's important to understand how animals move through space, but there's something really important about critical points or phase transitions, which is that there's ultra-sensitivity close to the phase transition point. So the brain, this even a little noisy brain of a fly, becomes like a supercomputer close to that critical point and can amplify any differences in the environment. And so this, again, is reminiscent of what we see in physical systems. If you heat up a magnet up to the Curie point, it can become disordered. The thermal noise spreads across the whole system. Now, if you look at the order of the system, the magnetization of the system, close to that critical point, close to that transition point, the magnet becomes incredibly sensitive to external magnetic fields. So if it's very cold, it's really hard to flip these magnetic spins the other way. You need a lot of energy to force them in the other direction for a cold magnet. For a very hot magnet, there's so much disorder there that you need a really strong magnetic field to convince them to move, to point in the same direction. But close to the critical point, the magnet becomes infinitely, well, for an infinite system, infinitely sensitive. In a finite system, it becomes extremely sensitive to external fields. Now, so here you need a very strong magnet to convince the system to change. Here a very strong magnet, and here tiny external influence can cause the spins to change. And it's the same process. We know that from analysis of phase transitions, that this ultrasensitivity is an inevitable property near the critical point. So in the case of the brain doesn't need to be tuned to criticality, geometry is an inevitability, and you get this emergent ultrasensitivity, so the neural activity that's normally just inhibited by, there's just so much noise in the system that the system cannot distinguish, suddenly close to the critical point, it's very, very sensitive. And it doesn't matter the angle that animals come in towards these two options, there's a manifold here at which the brain has to undergo this phase transition, or phase-like transition in the finite system. Okay, so how are we going to test this theory? It's going to be really hard, because if the brain is undergoing an ultra-sensitive phase transition, precisely to amplify information from the environment, then anything in my lab, like an air current or a light gradient that I can't detect, is going to break symmetry in the brain. So to detect this, we need to look at animals in an ultra-symmetrical environment. And in our case, we developed a virtual reality environment. So this tape does not have a volume above the table. I can tell you that right now. But even though you're complex humans, you still see it as 3D, even though it never was. So your brain is just tuned in to pick up on these cues. So we thought, well, if humans can fall for this illusion, surely animals can fall for it. Similarly, this shoe isn't actually three-dimensional. And so we can create a virtual fish, for example, and then we reconstruct the world 100 times a second from the perspective of the real animal, such that this virtual fish is just moving in a circle, but the real fish, zebrafish, believes it to be in the tank with it, volumetrically in the tank with it, even though the projection surface is the surface of the bowl. Now, we can't put more than one fish into our experimental arenas, because remember when the paper was moved ever so slightly, illusion was broken? It has to be precisely for one individual. But what we do is we network them together, so this fish interacts in real time with the hologram of this fish, and vice versa. And of course, we can control the topology of the communication, we can introduce time lags, we can have virtual fish, virtual predators, whatever we want. We can also play around with the laws of physics. I don't have time to talk about it today, but they do not represent space in the brain in Euclidean space, but in non-Euclidean elliptic space, which is super important for decision making. And so here you can see two fish interacting, not in the same world physically, but in the same virtual world. And we call this the matrix, because it's similar to the principle in the movie. And here are four fish interacting in the matrix. And because we're interested in general principles of how the brain represents space and time, we also started working on fruit flies, and coming back to the animals that I started with, also the desert locust, and it's on a big motion compensating ball. So let's test our predictions here. Remember, we had the prediction that as the animal moves through space, it should exhibit a bifurcation in the brain, which should be seen as an abrupt change in the trajectory. We could not find any paper, not one in the whole world, that anyone has recorded how an animal moves when it makes a decision. It's almost as if people think that animals look at the options, make a decision, and movement is just a trivial output. But in our theory, the movement is inherently important because it's this embodied process. So we conducted experiments with hundreds of fruit flies. These are the trajectories represented as the animal is flying through virtual space towards two static targets. They're attracted to vertical objects to perch on them, and clear evidence of the bifurcation. Similarly, desert locusts are attracted to vertical objects because they could be food sources, and the animal moves through this space and exhibits the bifurcation as predicted. Now, people often ask me, well, is this just a representation in the little insect brain? Well, we started doing experiments with humans, and these are unfiltered, just raw data from a couple of weeks ago. Potentially humans are also exhibiting a bifurcation as they move through space. Things get even more interesting when you start thinking of 3 or more options. The model, by the way, works for any number of options. What it predicts now is that the brain spontaneously breaks down the complexity of the world into a series of binary decisions, and always binary. Again, something we can prove in the analytic model. And so now, if the brain does this, why is it doing it? Well, again, at each critical bifurcation, the brain will spontaneously, due to this ultrasensitivity, remove the worst option. And then the next worst option, and then the next worst option. In our actual experiments, the options have to be super identical to each other to allow us to see the bifurcations in the brain. But the reason it's there is to deal with uncertainty or complexity of the world. And so we tested this experimentally, and indeed, as predicted, we find evidence of that the brain is reducing this complexity into an emergent sort of graph, effectively. And the space is continuous. We're not telling the animal to move in any way. We're just releasing the animal, and it can go wherever it wants. But there are these bifurcations that exist. And again, we tested this recently, again, unpublished data with humans, and I was kind of surprised. Potentially, this type of algorithm also works even for higher organisms like us. Okay, so I think this is a principle of decision-making. We've known for a long time that if it's easy to see a difference between options, you can make quick and accurate decisions. If it's hard, you can integrate over time. We heard this from Ahmed earlier today. These are both true, of course. But in addition to this, we argue that geometry, which has never been considered before to our knowledge, is also really important. Like, if this is a preferred option and this is a non-preferred option, a biologist would often just put the animal in between. Why on earth would it make any difference? But we predict it should make a profound difference. So working with undergraduate Lisa Ecker, we made a difficult challenge for the locust. They could only slightly prefer it when the brain couldn't go through the bifurcation. They were beyond the bifurcation point. But if they had the same distance to the options, but the brain was allowed to go through the bifurcation, again, as predicted by the theory, a massive amplification in decision-making accuracy. Again, as predicted by this theory. I mentioned before that the brain actually encodes information, encodes this vectorial information in a non-Euclidean way, in an elliptic way. That turns out to be absolutely critical for making decisions for very large numbers, or in fact, for 3 or more options. Is there a question?
Yeah. I was just wondering, in all these heat plots, what is this something in the brain, or is it the amount?
No, these are the trajectories of the animals.
Okay, so
we have hundreds and hundreds of trajectories.
How do you link that to the brain?
Because the brain predicted that there should be a bifurcation. The model predicted there should be a bifurcation here and a bifurcation here.
So it's the model that predicts, it's nothing...
The model predicts it.
And the model is the brain, or what?
And the model is the brain, that's correct. So the model made the prediction that we should see a bifurcation here and a bifurcation here, and then we run the experiments, and lo and behold, we see the same bifurcations as predicted by the model in trajectory space. The prediction, the model here is a model of the brain. And so if we run this model thousands of times, the model predicts that the animal trajectories should exhibit these bifurcations, each bifurcation associated with a dynamical bifurcation in the brain. So we should see these bifurcations in the trajectory space, in the space-time space, as the brain moves through the space. So as the brain moves through here, we predict it exhibits a bifurcation here. And if it chooses this way, we predict it exhibits a bifurcation here. And then to conduct these experiments, we run hundreds of experiments where the animals have the same options, and we analyze those trajectories, and we find evidence of these critical bifurcations as predicted by the model. Does that make sense?
Yeah, I just don't still understand how, like, so it's a model of a brain, but then it's the model, there's
So,
not,
so maybe maybe it's
where
the
is the
term
evidence?
bifurcation that's confusing here, because the bifurcation in the brain is a dynamical bifurcation. It happens to associate itself with a real bifurcation in space-time. But the bifurcation, so the animal is going through here, we predict the animal's brain is in a certain state. Here we predict the brain undergoes a bifurcation, a dynamical bifurcation, that results in an abrupt change of the trajectory. So this is the prediction of how animals should move through space with these three options, right? We then conduct experiments with hundreds of animals going through space, and we analyze the trajectories to see if there's evidence of these bifurcations in space-time.
I think the question is if there was neural recordings to validate the model, per se.
Yeah, that would be great.
Okay, I think that was the...
Yeah, I guess, like,
Yeah,
I wasn't
you're
like,
not recording.
no, okay, so it's a model of the brain.
Yeah,
So it's not the brain.
Well, I don't have the data here, but I can show,
Okay.
I can share with, it is a model of the brain. It's a model of the brain in the animal. Animals have brains. But we actually do have the neural data. It's supportive of this, but we haven't published that yet. But that's a collaboration with neuroscientists.
Yeah. Okay. Can your model predict the critical angle?
Yes, exactly. So what I was getting to here is actually the critical angle is the reason that we know that the animals are not using Euclidean space, right? And the critical angle is really important because there's only one warping of space that both matches the neural data. I don't have time to go through the neural data, but also the behavioral data. And it also is exactly the angle you need to make decisions for any number of options, or else you get stuck in endless loops. If the world gets complex, there's only kind of one sweet spot that works for all of the. So the theoretical data we have is now supported by experimental data, and which is now supported by neural data. But it's not that surprising to me because it's kind of the only way that it could work.
So, Iain, when you say there's this elliptic geometry, where is this geometry supposed to be living?
So it's in the ring attractor network. So this gets into some details that maybe it's more
perhaps
suited
I'll ask
to
you
others.
later.
What animals do is they either convert things to a cosine. The great thing about converting things to a cosine in the brain is you can add cosines. That's adding phases. It allows you to add vectors together. It can allow you to work out where you are, for example. And the neuroscientists like Larry Abbott call this weird because it's not a cosine, and it's exactly the functional form you need for what I'm talking about, which is making decisions. You then want to convert that to a cosine so you can do maths with it, vector algebra in the brain. So all of this is supported by neural data. I just don't have time to talk about that today.
Thanks, really nice. So the ring attractor in the brain is 2D, but I guess in real-life fish move in 3D environments. Does your model work in 3D?
It does work in 3D, but if you look at brilliant timing, if you look at the data from the fish, they always school in 2D, always. These are juvenile zebrafish. They're about 25 days old. They half a centimeter up, half a centimeter down, they're decoupled socially. Whether that's a constraint or a feature, I don't yet know. But it's a great question. We know that adult fish do not have this constraint, but perhaps luckily for us, these fish do. But the model works in higher dimensions. It works also in n-dimensional non-Euclidean space.
So just quickly, is it like a utility maximization kind of a thing?
No.
Or is it like a sequential decision making?
It's an emergent property of the dynamics of the model that breaks the world down into this series of bifurcations. We didn't tell it to do that. It's an emergent property, right? So if you imagine the local excitation and long-range inhibition, it's a bit like opinion formation in humans. If you're sharing opinions only with those that are similar opinions to you, and you're repelled from those with other opinions, then what we can also prove is there's only one stable bump on the ring, right? And that's good because the brain doesn't want to have two competing bumps because then you're stuck. So they're in this sweet spot that allows them to process information, in a sense, optimally in these environments and to make very accurate decisions. There's only two parameters in the model. In fact, we took the same model, exactly the same model, parameterized for Drosophila and Locust, and applied it to fish schooling, where now we have two moving targets. They're not static targets anymore. And I could never persuade real fish to swim exactly at the same lateral distance, but we predict as we change the lateral distance, the fish should exhibit a bifurcation, and indeed that's what we found. And similarly, if we didn't have the theory, we wouldn't know what experiment to do. If we did do the experiment and we got data like this, without the theory, we wouldn't understand it. But this shows you the double bifurcation. Just now it looks different because we have a moving frame of reference.
So you were talking about how that the brain has one essentially like bump, like has
Yeah.
a little place. Does that cause any tendency for like left or right-handed decisions where it's like, do you always go left?
So we have another paper that we're working, two papers. One that's actually you can see online, it's on the archive with Ben de Beauvoir, where we look at in flies. But we have another one that actually shows, and this is weird because this fits the neural data, that asymmetric networks, so in the model that I show you today, the model is completely isotropic in terms of its connectivity. And I thought that's great, right? Because you want to be spatially invariant. We find that the asymmetric networks create intrinsic biases like handedness. But they're arbitrary. Because it's a network property, you can change something like temperature or light levels, and the animal could reverse its bias. And this is what people see experimentally, and they can't explain it. But what's really counterintuitive is then, when they have a decision-making paradigm, they're better at making decisions. And that's what we still want to try to understand. Why is the slight asymmetry allowing, in network connectivity, allowing better decision-making? And it's not just that they break the symmetry or anything trivial like that. So it's a really good question, and maybe next time I could address that, but it's an important point. And another thing we're doing now is working with Arm and Bal, we are developing a virtual reality system where the animal is in virtual reality, it is fictive, it is paralyzed, but we're recording from the brain and we can decode when it thinks to turn left, thinks to turn right, thinks to accelerate. So now we can get whole brain imaging at cellular level resolution while the animal is interacting with others in the other tanks. And so this is pretty exciting. And if this is, such a general principle, maybe we could even see evidence of it in wild animals. And we saw this actually earlier today with Denai's data with the birds. I don't have that here, but if you actually look at the baboons in the brain of the individual, again, not within the brain, sorry, if you look at the decisions made, and you have this as a principle that we're looking for, we can ask, when some go one way and some go the other way, is there evidence of this bifurcation? And if there's an asymmetry, do we see them choosing the majority? And as Denai showed this morning, indeed, we do. So it does seem to be a very generic and robust finding in decision-making. And we've still got loads more to do to test, because we can make specific predictions. In virtual reality, we can hold the animal at the phase transition and test if it's infinitely sensitive, for example, or highly sensitive. And we've also done lots of other tests. But if I've got time, I want to talk about how we apply this to a major real-world problem. And it comes back to the locusts that I talked about at the very beginning. These are massive plagues, and they're actually getting worse, not better. And one of the reasons is that no one studies them, because they're basically impacting people who can't afford agrochemicals. So as far as you see, these locusts, they don't have wings at this stage of their life. They're marching in unison. And the FAO estimate the impact of 1 in 10 people on the planet through food shortage and contribute to emerging humanitarian crises, yet there's about 4 or 5 labs around the world studying them. And we were sort of kicked into gear when Martin Ensing wrote, even after 50 years, fighting locusts is more of an art than a science. And I showed you at the very beginning of my talk that what we thought we knew about locusts is almost certainly wrong. So really, there's a sort of call to arms here that we really need to bring new ideas to the table. And so what is the mechanism that people thought was coordinating it? Well, as I said at the very beginning, it was thought to be this explicit alignment mechanism from the Vicsek model. And I showed data that suggests that might not be the case. But why did people think this? Well, if you look at what's called the optomotor response, it's an innate reflex response. Animals cannot help but do it. If you show them moving dots or moving stripes, in this example, it's a fish, they will move to follow the dots or the stripes. It's a reflex behavior. It's very robust. If you look at this review of fish schooling, an innate optomotor reaction, a following reflex, ensures that fish maintain a single school during movements and rapid maneuvering. So people assumed, because you see this, and because it's a reflex, and because it's innate, it has to be the mechanism coordinating collective behavior. But they never tested it. In fact, in this paper here, they just assumed that the moving dots are sufficient. The poor locust itself can't even turn. It's fixed in place. The assumption is so intrinsic to how people think of collective behavior that they don't even allow an alternative hypothesis to exist.